Good afternoon, everyone, and welcome to the Flagship 3 session of our PERC Symposium. I am Mark Eramian, one of the co-leads for Flagship 3 of the PERC project. We're going to move right away into the pre-recorded videos for this session, after which I'll introduce our panelists for the ensuing discussion. Enjoy the talks.
Greetings all of you. Today I'm going to talk about data mining and machine learning approaches for data analysis, including hyperspectral data. So my name is Sindhuja and I'm from Washington State University. Although I would not call myself a machine learning expert, we use some of these tools for data analysis. So in Washington State, one of the major strengths is the number of breeding programs. So we have 15 plus breeding programs, which include cereal grains, legumes such as chickpea, pea, lentil, as well as horticultural crops such as potatoes, apples, pears, peach, to name a few. Many of these programs are supported by the commodity commissions. Below here is just a few to name, such as Washington Potato State Commission, as well as the Tree Fruit Research Commission. When you look at the strengths of Washington State University, it's not just the programs and the funding for those programs, it is the experts in different areas. So we have researchers working in genetics, genomics, phenomics, and bioinformatics. We have several scientists and researchers supporting these activities, the breeding activities to be specific, which include plant science, horticulture, plant pathology, physiology, soil science, computer science, to name a few. And finally, the excellent group or cohort of undergraduate and graduate students, postdocs, and technical supports. When I think of phenomics, I refer to the sensing techniques, not just that is high throughput, but also which can provide information with much better accuracy, which can be non-destructive, semi-automated or automated, resource efficient in terms of tools, technologies, as well as personnel, and finally is able to provide new insights of crops that is hard to predict. In my lab, we use different types of sensors, which includes handheld sensors, tractor-based systems, both for field crops as well as tree fruit species. The most famous one is the unmanned aerial systems or unmanned aerial vehicles, UAVs. We use several technology integrated with these platforms. And more recently, we are starting to explore high-resolution, low-orbiting satellite imagery. When you look at the satellite imagery, you can see that the resolution has increased with time. So previously the Landsat, which is 30 meter per pixel, and now we have PlanetScope and WorldView 2 with the best resolution of 30 centimeter per pixel. And this allows us to capture plot level data, which was previously not possible. Not just the spatial resolution, but also there is a better temporal resolution. You can capture and get data every 1 or 2 days. So these were just the platforms. We also use a range of different sensing systems, which include multispectral, hyperspectral, LiDAR, RGB thermal, alongside other technologies as well. So this is an overview of some of our more recent ongoing activities, which include the exploration of IoT-based systems for field applications, also volatile sensing, and also developing pipelines and data analytics, including app development as well. When I like to think about phenomics, there are three major pillars. One is the sensor or sensing system itself, which could be RGB, hyperspectral, multispectral, thermal, or any other type of sensor itself. The second aspect is the automation of the sensor to capture the data. And the third important is the data mining aspect. And I feel if you really want to make the best use of Phenomics technology, it is important that all these three pillars are robust and integrated with each other. When you look at the data processing, for example, here is an image, you do some pre-processing just to remove some outliers or other noises. You do segmentation, for example, you separate out your plants from its background. You extract features, for example, you can extract vegetation indices such as normalized different vegetation index or NDVI, or even color features or any other features. And finally, the classification. For example, in disease severity, you can classify the image based on the severity and symptoms. So these are some of the examples. And machine learning can be used in each of those steps. For example, in-weed machine learning tools, including deep learning, has been used for segmenting the heads, the wheat heads from the plant background. Segmentation, you can separate out your plants from soil in a good way using machine learning tools as well. In addition to that, you can also use it for image acquisition. For example, intelligent sampling, not per se deep learning, but other machine learning tools can be used for doing so as well. I just wanted to mention a couple simple examples over here in this slide and the next. In this slide, I just wanted to show, here we are trying to separate out the soil background from our plant of interest, and we are using color thresholding. And this is a k-means clustering, which is an unsupervised machine learning technique where you separate out the soil from the plants. And you have to be careful with the same laptop, the processing speed for the two techniques can be very different. But this is an era of edge and cloud computing. Hopefully things will improve with time in terms of demands using the computational power. This is another example where we did thresholding to separate out the canola flowers from its background, and also k-means clustering to do the same things. And we found that both of them produce similar results with very minor differences. In general, the color thresholding needs time in early parts of it where you need to establish your threshold. While k-means clustering is a little more automated, but when it comes in terms of processing time, k-means clustering may take a little more time than color thresholding. In this slide, I wanted to talk about RGB imagery. So here is an example of root rot disease in lentil. You have roots, you have the shoot portion. We extracted 87 features, which include geometric, color, and texture features. And you can see that if we extract each of these features from 3 regions of interest, it would be 261 features per sample. So if you have 1,000 lines with 3 reps each, it can quickly grow into 783,000 features. This is where we need machine learning techniques. Here is another example, but this is associated with the hyperspectral data, which was acquired from a spectroradiometer. And if you really see, even if it is 1,000 features times 1,000 features, 1,000 plus features, we are trying to extract the NDSI, which is a combination of every features that is available through the hyperspectral data. And each sample then is going to have 21,000 plus features. So if you have 21,000 features with 1,000 lines and 3 reps, your data size can quickly go into 32 million features. Again, this is where we surely need machine learning techniques to study the patterns and trends within the data set. I'm going to tell using an example of fire blight. This fire blight disease is a major disease in apple of great commercial importance in Washington state and other parts of the country. And if you really look at the phenotyping principles, these phenotyping principles refers to the standard way of phenotyping. At least 5 out of 8 steps can be done more efficiently using the sensor than evaluating the phenotyping approaches manually. So the sensing can help in eliminating the need of expertise to a little extent, can increase throughput, avoid personal fatigue, can be cost and efficient, and also provide a standardized way of capturing the data. So we collected hyperspectral data for 2 years, 2016 and 17, and I was just happy to see a similar pattern between 2 days, which sometimes doesn't happen. And here you can see that these colors are common across. So the data set, the hyperspectral data set, has an issue of collinearity. So several of the spectral features can be highly correlated with each other. That's why you see these patterns, similar patterns. So that's why you need machine learning to remove or understand some of these aspects of collinearities and look at clear trends. So the graph itself is a NDSI, so it's a combination of two wavelengths and its correlation with the disease severity. And in this study, we used two models. One is the quadratic support vector machine and PLSR, partial least square regression, to see if we are able to classify the resistant and susceptible diseases into two classes, which we were able to achieve. And mid-period and end-period refer to the middle of development and maximum development. And some of these changes happened. There was a lower performance during end of the seasons because that was also the peak stress period in the. We also use PLSR, partial least square regression, to look at or identify the NDSIs that were well representative of the disease severity. And this is an example of some of the NDSIs that were selected using the algorithm. This is another study which I wanted to talk about. This is a root rot disease, which is an important lentil disease. Mainly, it affects the roots. And based on a previous study, we found that the roots were the main, the symptoms in roots were much more prominent than shoot, especially associated with the severity symptoms. So we had two data sets, root 1 and root 2. Root 1 included all the samples, while root 2 eliminated the border scores, visual scores from the sets of samples. In our previous work, we compared the generalized linear model with elastic net, and we recently compared it with convolutional neural network, and there was big differences between the not big differences in the approach. In terms of results, they were very similar. So we thought, rather than having an input on the features that were selected and have a classification of different classes of diseases, convolutional neural network would be a little more automated. But of course, I should say there was a human input in terms of tuning the hyperparameters and also finalizing the architecture. When you compare the performance of elastic net and CNN, always the elastic net slightly performed better than the CNN. So here you can see the difference. The green ones are the root 1 and the gray ones are the root 2. And of course, the root 2 is a cleaner data set So that's why the performance was much higher than the root 1 dataset itself. So both CNN and ElasticNet were able to classify the resistant classes much better than the partially resistant and susceptible. In general, the partially resistant was very challenging because the features overlap. So with that, I would like to summarize by saying that sensing integrated machine learning can be a powerful tool in phenomics. You need good big data sets and optimization may be critical. And always more data does not mean better data. You need a representation of the variability in the data set. And understanding the science behind this data set is also important. With that, I would like to acknowledge my past and current students as well as postdocs, as well as the funding agency, USDA NIFA, as well as College of Agriculture, Emerging Research Issues, a bunch of breeders, and here is my contact information. Thank you.
Hello everyone, welcome to my presentation. My name is Hao Song. I'm currently a PhD student in Department of Computer Science, University of Saskatchewan. Today I'm going to talk about my study on plant root occlusion prediction using deep learning. Phenotypes are the organism's observable characteristics result from the interaction of its genotype with the environment. By studying the relation between genotypes and phenotypes, we may improve the breeding process by utilizing the nutrition and water supply. We're interested in plant roots because they are responsible for supporting the plant's above-ground structure and responsible for transporting nutrition and water. To study the genotype-phenotype relationship, we have been developing an automatic pipeline. The pipeline takes a set of hydroponic root images as input, process them with a set of operations, and then construct them into the 3D model. A set of root phenotypes can be directly measured from the 3D model or its abstraction model. So in today's talk, we'll be introducing one of the operations called root gaps implanting. The dataset we're working with is provided by GIFS. The roots are planted in gel tank, placed on the power rotation stage, and imaged are taken using an RGB optical camera from over 180 degrees. There are studies showing that the root in gel doesn't grow as well as in the soil condition. Thus, they added a supporting system to the gel tank to help the root growth, as shown in the right image. One problem of the dataset is part of the roots are occluded by the supporting system. Thus, when we apply segmentation algorithm, multiple gaps may appear, as shown in the red labels in the image. Before sending them to 3D reconstruction, the gaps need to be completed. Traditionally, the work needs to be done manually by sketching the missing root, which is very complicated and time-consuming. In this project, we proposed an automatic root gaps implanting method using deep learning approach. Due to the limited number of ground truths and dataset, we decided to train our model with a public synthetic root dataset proposed by Lobat et al. The dataset contains 5,000 dicode images and 5,000 monocode images, with variable size from 200 by 1,500 to 1,000 by 3,000. Our model is a generative adversarial network, which extends Chen's work published in 2019. The model consists of 3 parts: a generator, a local discriminator, and a global discriminator. The input images are first augmented and then break into patches. The patches are considered as ground truths. Then some gaps are manually added to the patches to create corrupted patches, which is used as input of the generator. The generator is responsible for the implanting work. The generator is a UNet-like encoder-decoder CNN network, where the images are first downsampled and then upsampled. The last layer of the model is a softmax layer, and the output is a probability map, where higher probability means it's more likely to be the root. The predicted patches are evaluated with the ground truths in the local discriminator. Also, the predicted patches are concatenated into a full image and then compared with the augmented root in the global discriminator. This step ensures the completeness and the root connectivity in the full image view. The final loss is a linear combination of local discriminator, global discriminator, and the generator loss. We have tested our model on both synthetic and the real maize root. Here is the result on the synthetic root. The model works really well on the decode root. It gives promised prediction. But for monocode, when there's high density and overlapping, the program may fail or give inaccurate result. For real maize root, the model works similarly. Our model has a great potential of predicting missing root when there's less density and overlapping. Here is a full image view. We can also say the model is not able to predict the vertical gaps or where the gaps are too large. Thus, our future work is to increase the training data size and the variability of gap shapes. We may also train the model with the real data in the future, so our model can be more adaptive to our real dataset. Thanks for watching. You can send me emails if you get any questions.
Hello, my name is Tewodros Ayalew, and I'm here to present a project that was done in collaboration with Dr. Ubens and Dr. Stavnes, titled Unsupervised Domain Adaptation for Plant Organ Counting. This work has been published in the CVPPP workshop that was held with ECCB 2020. Object counting is an important task in computer vision, which is applied to different tasks such as crowd counting and cell counting. It's also a highly relevant task in image-based plant phenotyping, where we apply it to count plant organs such as leaves and spikelets. And convolutional neural networks provide state-of-the-art performance for the object counting tasks. However, they require tedious annotation to train the models. Even if we have pre-existing data sets to train counting models, there is a common problem that usually hinders the performance of a trained model called domain shift. Domain shift is a difference in the distribution of the training data and the test data. For instance, a wheat spikelet counting model trained with indoor images would require fine-tuning if it's going to be deployed for in-field spikelet counting. So in order to solve this problem, we proposed a method, an unsupervised domain adaptation technique, to jointly train a convolutional neural network to count objects from images with dot annotations and adapt this knowledge to related sets of images where we don't have labels for. For that, we proposed a domain adversarial neural network for density estimation. So the whole goal of this network is to get domain invariant features that would help us count plant organs in the source or target domain image. To evaluate the performance of our method, we experimented on two separate counting tasks. The first one was a wheat spikelet counting task, where the source domain is the acid dataset and the target domains are crop quant and global wheat head datasets. And the second experiment was a leaf counting task. The source domain we used for that is the CVPPP dataset, whereas the target domains are Komatsuna dataset and MSU PID. We compared our method in the wheat spikelet counting task with baseline model that was solely trained with the source domain data, and then it showed that our method outperformed the baseline model. You see from the sample results, our model performs much better than the baseline model in counting spikelets, and on the second data set, our model actually outperformed the baseline model by far. As you can see, the baseline model misses most of the spikelets. We compared our results from the leaf counting task with a previous domain adaptation work and our baseline model. The previous work was trained with two separate loss functions, where one was a least square error and the other one was a cross entropy error. Our method performed better than the least square variant from the previous work in the interspecies experiment, and it performed better than the cross entropy in the intraspecies experiment. Our method performs consistently well without the need to change the loss function in a case-by-case basis. As you can see in the figure, our adapted method was able to localize the position of leaves in the target data set. The previous work we compared our work with had an assumption that the distribution of the source domain and the target domain should be aligned. In our case, we didn't have that assumption. So to show we have disregarded that assumption, we created a composite image by sampling for random image from the Komotsuna dataset and stitching them together. And then we used the model that was initially trained and passed this composite image. And as you can see in the output density map prediction, our model was able to localize and count the leaves without the need to retrain it. In conclusion, we tried to address the performance problem due to domain shift in image-based plant organ counting tasks, and we proposed an unsupervised domain adaptation technique, which performed better than the baseline model. Thank you.
Hi, I'm Nazifa and welcome to my presentation on a novel technique combining image processing, plant development properties, and the Hungarian algorithm to improve leaf detection in maize. Agriculture is the backbone of many countries' economy, and it's important to ensure successful crop cultivations and plant breeding. Manual inspection is not only time-consuming but also sometimes destructive. Also, maize is a globally grown, one of the most important cereal crops. So we aim to reconstruct and predict maize plants' growth properties, topology, numbers and position of leaves, and their emergence from indoor time sequence plant images. We had an open dataset from the University of Nebraska-Lincoln with 13 different maize plants of different genotypes. Plant images were captured for 27 days with the visible light camera from 2 different angles. As methodology, these are the steps of our method that we will go one by one, but very briefly. However, anyone is welcome to ask about more details. Okay, so at first we removed the background from the plant image with the background subtraction method and then applied Utsu thresholding on the foreground to obtain the binary image. Then we calculated the excess green image of the plant pixels obtained from the first thresholding and thresholded it again, which ultimately removes any soil or top pixels that were not thresholded before. Next, we calculated the area of convex hull to know which view covers most of the plant area. It's clearly shown in the figures that the 90-degree image has more information about the plant than the 0-degree. Then we skeletonized the binary plant image and applied a traditional pruning where a weight of each edge was calculated and any edge that does not have any role in skeleton reconstruction have been removed. Next comes our one of the proposed method where we eliminated skeleton spars with heuristics and statistics regarding maize plant. Following are the heuristics we used in our method, and they are removing spars that are 1 pixel long, root area pruning with maize statistics, and by comparing last 2 consecutive branching points position, removing edges near the root that has been created due to top pixels or non-smooth segmentation boundary, and so on. I will explain only one of the heuristics here for the time constraints. For example, the statistic says that the third leaf color of maize plants usually becomes visible approximately between 10 to 14 days after emergence. Hence, we calculated the number of branching points starting from the topmost as maize has an apical structure and deleted everything after third leaf color. The next proposed technique is to apply Hungarian matching algorithm. Hungarian matching algorithm gives the best matching by calculating the costs. In this case, here is a day 10 and day 11 plant image, and the cost is calculated with the Euclidean distance between leaves. And we can see here how the matching shows that there must be a leaf here on day 10 which has been missed somehow in the process of segmentation or pruning. We calculated the recall and precision to evaluate our method and implemented a convolutional neural network with deep plant Phenomics tool, and also calculated the mean absolute loss and absolute loss standard deviation. The ground truth number of leaves was calculated by taking the maximum number of leaves between the two views, and leaf detection was done visually. Our method detected 1651 true leaves among the 1883 ground truth leaves with only 23 false leaves. We can see here how the matching algorithm has improved the results. After comparing time series leaf count and leaf matching with Hungarian algorithm, the number of detected true leaves became 1674 and 16 false leaves. We implemented few CNNs with different hyperparameters. The base DPP leaf counter model, after a few trials, resulted in a mean absolute loss of 1.9 and an absolute loss of standard deviation was 1.5. We can see that both with and without the leaf matching, our method has fewer mean absolute loss and absolute loss standard deviation. I'd like to mention here that it's just a comparative evaluation that we did to see how our method is performing. It's not a direct comparison of our technique with deep learning approaches. So here is our recall and precision, and we can see how the time series leaf count and leaf matching improved both of the recall and precision. So altogether our method achieves a recall of 90.8% and a precision of 99%, and we believe that this work would contribute in component-based plant phenotyping, studies of maize, and ultimately in improving food cultivation. Here is the references I used in the slides, and thank you for bearing with me.
Hi everyone, my name is Lassoued. I'm a research associate at the University of Saskatchewan. Over the past 5 years, I've been managing an expert survey project investigating the regulatory and social issues surrounding the adoption of novel plant breeding technologies, including genome editing. Along with these new breeding methods, modern technological applications such as drone technologies and data analytics present another way to boost yield and achieve food security. To gain insights into the challenges and opportunities around using, managing, and sharing agricultural big data, my team and I conducted a couple of surveys between April 2019 and September 2020 to identify key important data and digital agricultural technologies to the future of ag, benefit and barriers to the adoption of big data analytics, and issues around data sharing and data management mechanisms. Some key results follow. According to the surveyed experts, different types of data can enhance food security, with genomics data, farmers' metadata, and phenotyping data having the largest impact. Except for farmers' metadata and telematics data, most respondents signal that types of data, most of them should be open, given their positive impact on research transparency, food safety concerns, pest management, collaboration, regulatory compliance, and management of scarce natural resources. As for the enabling analytics for digital ag, surveyed experts identified computational decision aid tools along with sensors and digital communication tools as the most cross-cutting technologies for digital ag. Geographic information and geolocation systems that collect real-time data are also important field-based activities. Most respondents reported that planning and forecasting, the identification of problems, decision-making risk management, and productivity are all likely to improve in their respective organizations as they make use of big data. On the other hand, most experts identified the lack of qualified specialists in big data analytics and the cost of investments as the major barriers to the adoption of big data in their work environment. Most respondents would be sharing their own data, whether directly with others or to publish them openly, mainly for public benefit, professional gains such as scientific merit or partnership, or to maintain transparency. security concerns over the handling of sensitive or confidential data, risks of cybercrimes, and worries about intellectual property rights and expropriation of ideas are concerns that limit data sharing. Surveyed experts are willing to share their own data with partners like regulators, national and international seed banks, university breeders, research organisms, and online data repositories, but unlikely to share them with supply chain integrators and multinational seed companies. My future research will be mainly focusing on identifying the factors that drive development and commercialization of innovative agriculture technologies, specifically identifying decision factors to license a new crop variety and those factors that drive adopter choices and the specific information they need to make an adoption decision. Thank you.
Hello everyone, this is M Khaled Saifullah. I'm the system analyst at the Department of Computer Science, University of Saskatchewan. Today I'm going to talk about P2RC Data Management Repository and Portal, which is under P2RC Data Management Group. Today I'm going to talk about the overview of the project. So I will talk about the implemented features and our future initiatives. This project has been initiated by Andy Sherb, Chanchal Roy, and Kevin Schneider as a PIs. There are some people at the research team, and we are all at the development team. The goal of the project was to have a sustainable preservation, retention, and sharing of P2RC data and the metadata. The objective of the project is to develop a federated repository and publicly facing portal, and we want to adhere the FAIR data principle, which stands for findable, accessible, interoperable, and reusable. We have proposed a data lake structure where the data set will go through some zones, for example, landing zone, staging zone, refined zone, and archival zone. And in each zone, there would be some activities such as visualization, modeling, analysis, curation, and so on. We will try to communicate with industry partners, academic partners, and other stakeholders to maintain the agile software engineering practice, and we will integrate P2RC clouds into our systems. We have investigated different related initiatives such as NCBI. However, we found that NCBI is focused mostly on genome data. Another initiative is GEMS, but the data analysis tools is very limited to RStudio and Jupyter Notebook. FISH and GenPeas are other two ontology-driven systems. However, it is really difficult to turn specialized information into ontology. Google Dataset Search is another search engine. However, there is no support for the data submission and analysis. CCAN and DSpace are the libraries, but these are not well suited for the private data repositories. Based on what we have learned from the previous or the related study, we come up with the design principle of our system, which we named as Site Data Manager. The design principle involves multiple stakeholder needs, user-centric design, use important, must add value to P2 and so on. We actually defined our key concept, and in key concept involves research group, user, datasets, metadata, data template, software, and news. And the way we have designed our system is in a tree hierarchical way, where each research group would be the node of the tree, and each research group will have one or more data sets, one or more data template, one admin, and other people would be group members, and there would be research group under research group so that it can be designed. If we take an example of P2RC, then Site Data Manager would be the root of the tree, P2RC would be the children of the Site Data, all the flagships would be the children of the P2RC, and all the research groups will be under the flagships. Now, if any research groups need subgroup of projects, they can easily define that from that specific research group. Based on that design, we actually defined our abstract data model, which is built on ASN.1, and we try to define each of the entity of our design. Our implemented features involves login, which includes USask login and Globus login. We also implemented Elasticsearch based search and filter. We have Globus data transfer based data upload or the submission. We have chunky file browser with the image viewer attached, data visualization. However, we are working on different other visualization for other file types. Our plan is to have simple, easy to use, and interactive UIs for managing dataset and groups. And also we organize our data repository in such a way that provenance and versioning can be supported.
Here are some screenshots. At the top left, you can see the homepage of our system. At the top right, you can see the dataset list view after searching and filtering. At the bottom left, you can see the data viewer at the front and dataset details view at the back. And at the bottom right, you will see the group details view. We have also conducted some meeting with the flagships, and we come up with some requirement. For example, common requirements is crosswalk between different datasets with plot vision. Other requirements can be uploading to NCBI dataset versioning, cleaning and sharing, organizing Canola datasets into databases, integrating annotation tools, fast access and sharing of active data. Our future initiatives involves deploying testing and the production server for the users, communication with data analysis platforms such as Saifullah, supporting data transportation from data management systems such as NCBI or Nopals to data analysis portals such as Plot Vision, dataset versioning, and standard data and metadata templates. This is our demo sets, and I'm really happy to answer any of your questions. Thank you.
Good afternoon, and welcome again to the Flagship 3 session of our symposium. We will start the panel discussion in a moment, but first, in case there are those joining us for the first time, I'll briefly orient you to our virtual format and then introduce our panelists. All the sessions are being recorded, and the portal where you access today's session will be our landing place for access to links to all the live sessions and recordings after the symposium. If you're having technical difficulties, there's a blue help button at the lower right corner of the screen. You can click on that and then click Contact Us for technical support. There's also an email link to log in on the login page for the portal. Lastly, you can click on the question mark button on the web player that will redirect you to the help page. Attendee microphones in the sessions are muted. We do, however, encourage your questions and interaction with the speakers. If you have any questions during the session, you may utilize the chat box feature within the media player to submit questions and chat with the attendees. This is the button with the speech bubbles underneath my face. You can also click on the Ask a Question button, also located on the media player, to submit a question to panelists. This is the button with the hand on it beneath my chin there. Our team will be monitoring both platforms during the presentation and answering your questions, either answering your questions or communicating them to the speaker. So I'd like to introduce our panelists for today. First are the speakers from the talks that you just saw. Our keynote speaker was Dr. Sindhuja Sankaran from Washington State University. Then we had Hao Song, Tewodros Ayalew, and Nazifa Khan, who are computer science graduate students at the University of Saskatchewan. Also welcome to Lassoued, a research associate in the Department of Agricultural and Resource Economics at the University of Saskatchewan. And M Khaled Saifullah, a systems analyst in the Department of Computer Science at the University of Saskatchewan. Also joining our panel today is Dr. Carl Goodwin, the PERC Flagship 4 co-lead from the Department of Computer Science, and Dr. Andy Sharp, who is the PERC program director. And now I'm going to open the floor for questions and discussion. Looks like we've got some questions in the queue already. Our first question is from Ian Stavnis for Sindhuja. He asks, among sensors, automation, and data mining pillars, which do you think has the most potential or need for improvement for advancing plant phenotyping? Sindhuja?
Thank you, Ian, for the question. In my opinion, I think all the three pillars are important: sensing, automation, and data mining aspects. So I would say in terms of importance, I wouldn't favor one than the other. All the three are important. Without a standardized sensing protocol, it's hard to develop data mining approaches. Automation is also important. In regard to the need, I would say there is a lot of focus in the first and the last aspects, sensors and data mining aspects. Not as many in automation. If you really look at the automation systems, you can see that there are so many different types of automation system developed, mostly crop specific or cropping system specific. So I feel, and sometimes people just don't develop the automation tools. They they just focus on even handheld sensors or drone-based sensing systems. And of course, automation is possible with drones as well. So I feel that especially in regard to the ground-based systems, automation needs to move a little further than it is currently.
Thank you for that answer. Are there any other panelists who would like to comment on this issue? No? Okay, then I will proceed to our next question. Our next question is from Kevin Stanley, and this question is for Tewodros. Kevin asks, your method for, it was transfer learning. Your method outperformed the baseline, but he asked, were your results good enough to be useful? And if so, to who and how?
Thank you for the question. I would say yes. It can be applied to any organ counting task where we have a data set with a notation for a source data, but no annotation for the target data, and especially when there is an existing domain shift for tasks such as indoor to outdoor counting tasks, like the specific task we tackled in the paper was spikelet counting, where the source data set was annotated for indoor data, and we wanted to apply the knowledge that we got from trading in the indoor data to be applied to in-field prediction. So we applied it there. So it can be useful to any organ counting.
Did you say that you actually tried it in an outdoor setting as well, or you just think that it would work in an outdoor setting?
We evaluated it with an outdoor data set.
Oh, okay. So you didn't move your plants outside. You just used a data set that was taken outside as well.
Yeah. With different distribution.
Did that data set have all of the variation that you'd expect from normal imaging in the field, like different weather conditions and different times of day and stuff like that?
We tested it on two separate data sets that have different variation, but we didn't actually go through individual variations. But it should work because the procedure we took, the method, would account for that.
Is it possible to take your data sets and kind of simulate different environmental lighting conditions? Like, can you put a filter on that would be like a different sun position or cloud cover or things like that in order to do testing under different lighting conditions?
We have a plan to do that, like taking a simulated data with these variations and then adapting it to real world data. We have actually tried it, but we haven't evaluated it because of the lack of existing annotated data. But we tried it and it performs good.
Any other comments about this on this topic from the panel? Okay, then I will go on to our next question. Our next question is from Jordan Ubbins, and this question is for Nazifa Khan. And Jordan asks, did you compare your leaf counting method against the leaf counting function in Plant CV? He says they use maize in their examples. So, Nazifa?
Hi, Jordan. Thanks for your question. Yes, I did give a try with Plant CV functions. And though the segmentations was okay, but the skeletonization result was more better of our method, because there were a lot of extra spars and discontinuity in the skeleton from their skeletonization function. So I didn't continue towards that because the skeletonization step is one of the important steps in our study. because most of the pruning strategies are based on the skeleton we got, and it was better the less spur we had from the skeletons. Thank you.
Okay, any other comments or questions from the panel on this topic?
I've got a comment, just as a follow-up there for Nazifa. Nazifa, have you tried using the technique that you've developed on other types of crop data? Has it just been maize, or have you been trying other crops like wheat or canola, for example?
Oh no, at this point we just tried on maize, but with canola I'm working on another that's more like holistic phenotyping, but we do have a plan to move with the component-based phenotypic analysis of canola too. But probably the skeleton parts would be difficult for canola as it was more straightforward for maize. Mm-hmm. So at this point we were just focusing on maize.
Yeah, I guess that's going to be a key thing. The architectures of different crops are very different.
Yes. And the structure, because of the apical structure of maize, it was more easier to analyze this and get the skeletons from the side view, which is not the case for canola. So.
Okay, thanks.
Thank you.
Other comments? Okay,
I just have a question to follow up. I was wondering, would it work, or a modification, would it work under field conditions?
The same approach only applicable for the field conditions, because in field the plants are more overlapped or besides each other, which is not the case for when they are in the phenotyping facility indoor. So the process would need some modifications if we want to work on the field images, aerial images of fields.
Okay. And I guess I will move on to our next question. Our next question comes from Kesav Singh, and this question is for Sindhuja. Kesav asks, how is NDSI approach different and better than Wilks's lambda bands selection method?
Thank you for the question. We have not used the Wilks lambda band selection method, so I don't have a good comparison to state. But with the NDSI, we did find some positive results, not just in the reported study, but a couple other studies we had done.
Okay, that seems like a very specific question, but any other inputs from the panel? All right, I don't see any more questions in the queue right now, so I'm going to ask a question of my own to some of the panelists here that haven't yet had a chance to say anything. I'm going to start with Khaled, if that's all right. This might not be a great question because this is not a little bit out of my area, but could you perhaps elaborate on how you'll manage the metadata standards in the systems that you're building, and whether you've been able to give any consideration to the interoperability with metadata from other repositories and any associated challenges around metadata?
So thank you very much. It's a really a good question, because when it comes to fair data principle, when it comes to interoperability, then metadata is a big concern. So actually what we are currently doing is we are actually adapting a metadata template or data template for our PERC program. And we are actually looking into a different types of metadata initiatives. One is MAIAPI. I think it's a community standard metadata initiatives, and the definitions there are really broad, and that can actually help us to define the metadata for plant phenotyping. experiments. Also, we also look into different initiatives, for example, doubling core metadata initiatives, which mostly describe the general metadata, and a lot of other schemas and ontologies are also used, doubling core metadata initiatives. So in short, I just want to say that we are actually implementing that metadata template for PERC, and we will try to confirm that metadata templates or the data templates will follow that FAIR principle, so that our data sets or the data in PERC can be interoperable in different systems and platforms.
Can I ask a follow-up, Mark? I'd like to know, how has it been so far gathering data from PERC researchers? How difficult is it to put it into those correct formats?
So far what we have understand, or by talking different flagship leads, different research groups, we have understand that there are some similarity between how the PERC data has been collected so far, but yeah, however, there are a lot of practices going on in different research group. So we have collected different resources, different data from different research group, and we found that if we start the process of using a standard template, data template or metadata template, it will not be that hard. But yes, it's true that the way that research data has been collected so far in PERC, there are a lot of variations, and we are actually trying to make a bridge between each of the data templates, so that we can support any types of data within any types of PERC data within our system.
Further discussion amongst the panelists?
So Mark, I just had a follow-up comment. So isn't it very challenging to adjust, like to have a metadata of the phenomic data where there are so many factors that tend to affect? There could be a wind, changes in weather patterns, weather conditions. Are all those data go into consideration? And also the setting. And even if you buy the same sensor a year from now, something in the sensor itself has changed, right? So the companies are always working to improve their systems better. How do you incorporate it in your data mining?
So about managing the data, or about the in terms of managing the data, I'm not talking about mining the data. So in terms of managing the data, the system we have built is sort of it's a collection of different microservices, and you can easily our design principle is you can plug in any types of microservices into the system, and that will be integrated, that will be a part of our system. So we always feel that we always need to be adaptable there. However, what we are trying to understand is what are the use cases for the researcher, for the data scientist or the data generator, and how we can implement that in a fair using the fair data principle, or how we can ensure the fair data principle based on their user stories or on their requirements. So that's where we are actually working on. And also, as I have said earlier, since our system is something called a collection of microservices, different types of services. So like for the data templates, we have that notion that you can define your data templates or metadata templates, let's say in future, and then you can use that updated metadata templates for the new data sets. Also, we are trying to incorporate the data set versioning, which is also include data template versioning, metadata versioning, also the data itself versioning. So all of these things are in the queue in our development, so that, again, I'm saying any types of adaptability will be possible in future.
Sorry, probably I mistook it. I thought you're trying to generate a standardized metadata format for all the different types of data.
So yeah, this is our, currently what we are trying is standardizing a metadata template or the data template for PERC. But I can't ensure that this metadata template will work after 6 months. Maybe there might be some new things. So that's why we are saying that, yeah, we will have that versioning thing, so that within that metadata template, even you can, as a research group, a user can actually create a new version of that data template and use that for their purposes. And this is how we are actually planning to work, and we are integrating, implementing in that direction.
Yeah, maybe I can also comment as well. Absolutely, we sort of see things. It's evolving all the time in terms of the technology, and therefore the data that's generated and the metadata that's generated. So it needs to be kind of something that's monitored and developed as you go along, kind of like living standards, really, that need to be implemented. And to make this work properly, we need to have very good communication between the data management group and the research group. So that's one of the key aspects here, is maintaining good strands of communication between the different groups so we can properly support the initiative as we move forward in phase 2. So we've got those connections established, and we'll be certainly be maintaining them and hopefully strengthening them as we go on through the program.
Okay, any more comments? All right, then I would like to ask one more question of my own to get one more of our panelists involved before I return to questions from our audience. This question is for Rim Lassoued. One of the take-home messages I got from your talk is that you identified some challenges in creating and sharing big data, such that there are certain types of data that are in demand, like data from like farmer metadata, but that this data is less likely to be shared for various reasons. Could you talk about how you might incentivize the stakeholders who are holding this data, who are not sharing them very much, to maybe share them more?
Thank you, Mark. So just to clarify, with the surveys, we're not targeting just farmers, but mainly experts in biotech and also in social science. So among the concerns raised are privacy and confidentiality. So that's the main reason for farmers that they might not share their own data. And also the value of this data may be of a more benefit for those who will be using this data rather than the farmers themselves.
Okay, would you be able to say anything about, I'm sorry, I'm not sure if you were done with your answer, if you cut out. I would just like to follow that up with, you've identified the problem there. Is there any way to get around those reasons why that data is not being shared, to incentivize the sharing of that data?
Well, the problem is, if people will be sharing data, like what are the kind of benefits they can enjoy? Like for farmers, if they will be providing their data like to multinationals, it's like there is a question of trust. So we ask the question of trust, and it looks like banks or financial institutions and also consultancy agency are the least trusted compared to universities and government. So about the incentives, I'm not sure like what kind of incentives. That's a good question. We might investigate that in the future. What makes like different stakeholders willing to share their own data?
Okay, thank you. Any other follow-ups or comments from the panel? All right, hearing none, I'm going to return to our audience questions. I have another question from Ian Stavness. This one is for Hao Song. So Hao, Ian asks, what are the final targets for your root phenotyping? Is it segmentation? Is it skeletonization? Or is it more aggregate root traits?
Thanks for asking. So as I mentioned in the talk, this project I'm talking about is a part of a big pipeline, which take the images and start building a 3D model from them. And the final goal of this model is to measure root traits, which can be morphological root traits like root length, root width, and or it can be topological root traits like the number of branchings and the number of lateral roots or secondary roots. So we're interested to analyze the relationship between phenotypes and the genotypes, and so we can use this tool to give a good suggestion to utilize the plant breeding. So that's our final goal. And segmentation and skeletonization can be part of the abstract model. And the segmentation is actually one step before the inpainting. So we're starting from the segmentation and then we are filling the gaps occluded by the supporting structure. Yep.
Any other comments on that? That's pretty.
How? Where do your images come from of the roots? Are they taken from a lab system or using an existing data set, or do they come from PERC?
The images are from GIFS, and they take the image from the lab, and the roots are growing in the gel tank.
Oh yeah, okay. So you're not separating roots from soil or anything like that. These are sort of grown on a plate, right?
The roots is growing in the transparent glass tank filled with gel, and it's kind of like a hydroponic system. It's different from the soil condition.
Right. Do you think that it will succeed when you shift to, I don't know, doing X-ray imaging of roots in the field, or basically we can't always grow plants in hydroponic gel. So how are your systems going to scale up? I assume there's going to be a lot more environmental noise in the signal when you are using different imaging techniques.
That's a good question. If we use X-ray imaging or other type of like a neutron or other like a rays, like it's kind of a similar process. We need to find the segmentation first, and then we reconstruct 3D model, and we can take the measurement. The pipeline I designed for this dataset is concentrating on solving the occlusion stuff, like the gaps. For X-ray imaging, it would be even easier because there's no such kind of like occlusion. But there's some justification need to be made because those kind of like the X-ray technique, the beams are passing through the root, and for optical images, the light are reflected by the root surface. So the way of building a 3D model may be different. Because for those kind of X-ray methods, we can just do filter back projection. That would be easier for the building 3D models.
Thanks.
Okay, maybe sticking with the theme of roots for a moment more, I have a question from Felix Akins regarding the root project that Hao is working on. Felix asks, do you believe you need to make the model more complex or simplify it? Because I wonder what aspect of the gaps really create the error term, so especially small gaps. It seems like you might already get good results by looking at the ends above and below and just draw a straight line, just estimating the thickness, but I'm not sure how bad that would be in practice. Okay.
Regarding to this question, I would say by manually sketching the missing part, it's kind of time consuming, and for very large images, it will be more like manual work. And for accuracy, it's hard. It highly depends on the people who do the sketching, because if the people with less of a biology and plant science knowledge, if they just draw a straight line, they may miss some features. So it's hard to just by guessing say like which one is better and how bad that would be. But in practice, just sketching lines should solve the problem quickly. But yeah.
So just if I may presume to interpret Felix's question, I think they might be asking not about manual sketching, but about developing a system to automatically find the ends of these groups beside the gaps and putting the straight lines in automatically. Does that make you change anything about your response?
Yes. It's hard to be automated, I would say, because with a relatively larger gaps, it's really to just by guessing like which part from the top should be connecting to the bottom, and if there's overlapping, there will be more error happening. So we want to leave this work to deep learning since it's more robust, and by looking at more samples, it may give a even better guessing than people. So I'm not sure if that answers your question, but yeah, I would say deep learning should do this work better and fast.
Okay, other comments? Just a quick additional root question here then from Karim Panjvani. What is the end benefit? What is the end target benefit of filling the gaps? Is it improved 3D reconstruction or more accurate phenotypes?
That's a good question. The first purpose of doing this project is to complete the missing data so we can have a more accurate phenotypes. For example, like by measuring root volume or biomass, if you're missing multiple segments, it's definitely there'll be higher error rate. Our estimation should be less than the ground truth. So by completing the missing gaps, sorry. So yeah, the phenotype should be more accurate, by sure. And I'm not sure how much it will improve the 3D reconstruction, because without the occluded part, we can still build a 3D model. But still, like from that model, like how do we measure the accurate phenotypes? Like we still need to find the missing information there.
All right. Then one quick follow-up again from Karim on that, and then we'll move on to a different topic. He asks, is there an estimate of how much the trait measurement can improve, or is there an estimate of how much the trait measurement can improve by filling in these gaps, I presume?
At this stage, I don't know how to estimate. I don't have a quantitative estimation on how much it can improve, but more specifically, like at least for root length and root volume and root biomass estimation, it should improve a bit, depending on how big the gaps are. If they are larger gaps, it will improve more.
Okay. All right, thank you, Hao. I'm going to go to another question from Ian Stavnis then. And this question is for Sindhuja. Ian asks, WSU, Washington State University, is located in a very diverse agricultural region. Do you find that working on phenotyping across diverse plants and crops is helpful for your research program compared to, say, focusing on maize and soybeans?
And the answer to it is yes. And that's the exciting part of working in Washington State. It's not just because of the diversity of crops, also diversity of breeding programs we have in the state. So it is very useful to the program. However, I would like to mention that each cropping system is very different from one another. So we are learning in the process. So even if we can use a drone-based multispectral sensing for different crops, the findings are going to be completely different. And we are the plot size or many different things are quite different between the breeding programs. So it's a lot of learning in the process, and we continue to learn through this experience.
Okay.
Just a quick follow-up there. What proportion of the research that you engage in is, in terms of the collaborative research that you do, what proportion is sort of academic and what proportion is with industry? Is it fairly even, or is it more one over the other?
I didn't quite understand, but like with the industry, most of our collaborations are with the breeders. So the breeders include Washington State University is a public institute by itself. In addition to that, we have USDA ARS labs, which are also public breeding programs. So we collaborate quite a bit with the breeding programs. There's just, in terms of private partnership, the only company we work with is a seed company, which does breeding on its own to develop better seed varieties. But mostly I would say it's public institute breeding programs. I won't call it academic because these breeders release varieties that the growers are using in their field. So I don't want to call it academic. They are closely working with the growers or farmers to release the varieties that are developed.
Right.
Can you tell us how close you've come to being able to make a difference in those breeding programs? Like, are the breeders that you're working with actively using your systems, and have they found any of this genetic gain that we were talking about earlier through your systems?
Yeah, that's a very good question. And in terms of breeders itself, some are more exploratory, some want to implement as a part of their program. So we have a wheat breeder, Dr. Aaron Carter, and he is using some of the drone-based multispectral imagery for his decision, especially in early generation of his wheat trials. The others are not convinced yet, but they don't want to discredit the potential of the technology. So I do work with them on some of the research programs, trying to see how things can be improved in a good way. In terms of genetic gain, I don't have a good answer to it. We haven't got that far. I should also say that not all the breeding lines, the traditional breeding does not genotype all the varieties, all the lines. So that is one limitation. So these are traditional standard breeders where they are just making these crosses and making their selections. Unless we do a genotype based analysis with the materials, it's hard to highlight the genetic gain offered through a technology.
Eastern Washington State is actually quite a bit like Saskatchewan, isn't it? You grow wheat and it's flat and it's dry, right?
No, it's not flat. We have rolling hills. Okay. And yeah, it's very different from Saskatchewan, but it's as beautiful though, especially in this time of the year. So.
All right, we're getting near the end of our time here. Looks like I get to ask a question at this point. And I'm going to ask this one to Tewodros. In your examples of domain transfer, I noticed that the target data sets didn't seem to have much difference in scale from the original data sets. That is, the wheat heads in the target data set were about the same size in the picture as the original data set. So I'm wondering, is change of scale an issue in domain adaptation, or are the techniques that you're using robust to scale changes? And if so, how is that done?
Thank you for the question. It's robust for the scaling. We have tested it in a different scaling, but we wanted to align to similar methods that were done in other works. That's why we used those patch size that were displayed on the slide, but it's robust with scales.
Okay, thanks. Any follow-up from anyone? All right, then I have another question for Sindhuja. I noticed that in your talk that you had a few examples of where you were using traditional image processing like k-means and co-occurrence matrices, and also traditional machine learning such as support vector machines. So my question for you is, how do you see the role of traditional image processing and machine learning in the context of the rise of deep learning? Are there benefits from using traditional methods in conjunction with deep learning?
So I'm just going to talk based on my experience in terms of deep learning. I still feel it's a big black box, and it has a lot of steps involved, though it has a lot of potential. I'm excited to read new outcomes of deep learning. For example, one of my students who was using the deep learning, and there is no good rule on how do we tune some of these hyperparameters, and how do you develop an architecture? Does it need to be deep or shallow? Or there's a lot of things involved in it. And I'm more a user of technology than a developer itself. So I'm just telling based on my experience. In terms of the traditional ones, I feel that the traditional ones are good for base analysis, but we should also be aware that data sizes are increasing. So if you have thousands of materials with thousands of different traits, we have to rely on some of these advanced machine learning methods to help us look at it. But I don't know, at least to my experience, I tell my students, yes, you get an output from a machine learning algorithm. It's always better to go back and look at it visually and see is things the way it should look like. So I feel it's a combination of both is going to be the right way. And some of these advanced methods are also, it's hard to implement expert opinion. An experienced breeder has so much knowledge. They are seeing things that they cannot describe in words. So I wish if we can integrate some of those, okay, we have to look at the tip which is curved, or it's a simple example I would say is we had a project where we were trying to identify between common and club wheat. So these are wheats. Club wheat is a little bulkier, and common wheat is thinner. The club wheat is the one which is used for cakes, and it has a premium rate associated with it. And a breeder knows even a club wheat variety has some common type looking seeds. So some of this knowledge, if we just rely on machine learning, we are not going to know that even if it's club, it's going to have common seed or common wheat kernel looking like seeds in them. So I would say combination would be the best.
Panelists? Then I'll throw in a follow-up. So Nazifa, you use traditional techniques as well in your work. Do you have any thoughts on this? Why was using traditional method image processing an advantage for you?
I will add that thing. To me, deep learning is more like a black box at this point. And for example, the leaf tracking or leaf detection architectures that are more like, okay, they say that these are the number of leaves in this picture. but to know precisely where the leaves are and what the curvature of the leaves and what is the angle of leaves with the stem, to more about the phenotypes of the plant, I felt that if we could use traditional image processing with plant models, or like as she mentioned, the breeders have a lot of information about how these things grow. So if we could get their knowledge to enhance what we are doing and to enhance our the techniques accuracy, I thought that would be good thing. So that's why we try to focus on how we can use the plant models information to traditional image processing, or even traditional machine learning, or even deep learning, because I know that Jordan did a work where he has used the synthetic images from plant models for training the deep learning. So this was my main motive, to use the plant models, plant related informations, how the plant grows, what is the attributes of the plants, how the plants looks like, the statics of plant growth, to add more with traditional image processing technique to more about phenotyping different phenotypes or traits of the plant.
Okay, thank you, Nazifa and Sindhuja. Those are some really interesting insights. Any other points to this topic? Well, then, while I've got Nazifa with her mic unmuted, one more question for you, Nazifa. Could you talk more about the importance of incorporating heuristics in your work that were specific to the maize plants you were working with, and how critical was that to get the successes that you had?
It was difficult at some point, because the skeleton spars were like if it was difficult to differentiate between a skeleton edge, that is a spar, or a leaf, or a stem part. And due to the segmentation boundary around the root, we were getting a lot of skeleton spars that we didn't want to miss any leaf, but we also wanted to get rid of the false leaves. So we had to add some heuristics, like how a leaf look like, and what are the features. For example, angle of the tip edge with the root won't be same as an angle with a leaf with its stem. So we had to heuristics like that to differentiate between spar or a leaf, just to get rid of a number of false leaves. And we have noticed that these heuristics has reduced a lot of number of skeleton spars, and also it was kind of a balance where we were able to get rid of false leaves, having a good number of true leaves detected, because in some way we were probably getting rid of false leaves, but we are also missing some true leaves, or we were having a large number of true leaves with a large number of false leaves. So the heuristics were a balance between these two, I would say.
Okay, thank you.
Thank you.
All right, looks like I will ask one more question from the audience. So this question is from Danny Huang, and the question is for Hao Song. Danny says, I like the idea of working in terms of patches to construct the full synthetic image. Have you tried experimenting with different loss functions, discriminator architectures, and generator architectures? If not, I think it would be a good possible direction to pursue in order to improve and build upon the existing root predictor. Hao?
Yep. Thank you, Danny, for the suggestion. This is already my future plan. Because we're having difficulty like with small patches. The current patches are we use two size. One is 64 and one is 256. And because our images are really large, they're like 6,000 by 4,000. So smaller patches may not cover the full gaps, as I showed in the result. In the bottom of the root, if the gap is very large, it's larger than the manually created corrupted gaps, then the model will not be able to predict it. So we're thinking to change the generator architectures and use larger patches, or maybe use like overlapping patches and variable size patches to try to retrain the model and make it more adaptive. And thank you for your suggestion. That's already in my plan. Yep.
All right. Thank you very much, Hao. And at that, we've reached the end of the time for our session. So I think I will close the session now. I'd like to thank all of our panelists for their participation today, especially Dr. Sankaran, our external keynote speaker, and all of the participants for their great questions. So now I have been told that I am to turn things over to Andy Sharp for some closing remarks.
Okay, thanks very much, Mark. Really appreciate you moderating this session. And yes, I'm just going to finish off this year's symposium with a few particular comments. I think it's been a really quite successful. This is our first attempt at a virtual meeting, a virtual symposium, and I think we've had some good sessions, good full sessions with representation from the different flagships. Also, of course, representation from keynote speakers from around the world, and certainly appreciate the participation from our keynotes in the symposium. I have a few thanks to go through. But first of all, I wanted to announce the winners of our poster competition. As you might have seen on the portal, there's been some posters represented there, and they've been reviewed. And we have some winners of the poster competition, and in no particular order: Masi Aslay, Sarah Mardan Nisamani, and Anjika Atanaki. I'm sorry if I've mangled the pronunciation on some of those names, but congratulations, you're the winners of the poster competition. As well, we also have a People's Choice Award, and that's going to Julio Torres Tello as well. So they're the four winners for the poster competition. So congratulations. There'll be follow-up post-symposium. So you'll be able to collect the awards that go with winning the poster competition. I want to note that the posters, as well as all the sessions, the recordings of the sessions, are going to be available for the next year on the website. So GIFS will be sending out instructions to all the participants in the next week, so you'll be able to access them. So you can, if you want to dip back into the sessions and just re-see some of the presentations, you'll be able to do that. Feedback from participants is key. This is our first virtual event, and I think things have overall worked pretty well, but I think some things could be tweaked as well. So feedback from participants will be essential, and I think we'll have a survey that will go out post-event where we'll be able to get that feedback. I'd like to thank Perform Media for providing the platform for this virtual event. I think things went overall pretty smoothly, so congratulations to them on supporting us in this. For the program itself and the development of it, we had a program committee. I'd like to thank Chris Barker, Stuart Smythe, Steve Shirtliff, Tyrone Keep, Naveed Basgali, Jyoti Mondal, Carl Gutwin, Kirsten Bett from the other GIFS or the university, and also Steve Hunt from Cubic Systems and Alan Grombacher from Nutrien for working hard on the committee to provide us with the schedule that we had for the three different sessions. I'd like to thank Alan, Naveed, and Chris in particular for being part of the poster competition subcommittee. So those are the guys that helped to review the posters and selecting the awards there. A big thanks in particular must go to Monica Gordon, who's been handling all the logistics for this meeting. She's at GIFS, and thanks Monica for working so hard on making this a reality, this symposium a reality. So with that, I'd like to finish by saying thank you to all the speakers and the moderators and the participants for taking part in this virtual symposium. I hope you'll all be able to return to the next one in October next year, the 6th symposium. And maybe it'll be face-to-face. I certainly hope it will be a face-to-face symposium, but potentially it might even be a hybrid face-to-face stroke virtual meeting. More information about the next meeting and PERC in general will be, you can find that on the PERC website. So that's ptirc.usask.ca. That is a resource which is being redeveloped at the moment. We've had representation, in particular the phase 1 part of PERC on there, and that's now we're rejigging things to represent phase 2. And I think it's one thing that we definitely want to improve and enhance in phase 2 is our communication strategy, our engagement strategy, not just with end users, but I guess with the public in general. I think we have an opportunity through the website and other forms of communication to really show what we're doing within the PERC, some of the really fantastic research that's going on, and our collaborative partnerships that we've developed and will be developing. So keep an eye out for the website and see how things develop there. So with that, I'd like to thank everyone very much for taking part in the symposium, and I'd like to wish a good afternoon or good evening or good morning wherever you may be, and look forward to seeing you next year. Bye for now.